Right now you must be able to see, right? Just click for the link again, then we are live. Okay, right now I think we are okay. Just let me know whether am I audible or not. आवाज आ रहा है मेरा पूरा? Hello. Click for the link again, then we are live. Okay. So we are starting on today's lecture. So today's lecture is about this DNA replication. Okay. So let's start with today's lecture. So let's in between this DNA replication, the first point we will have to see that is about the wait a minute. The first point which we are going to see is the replication in case of the prokaryotes. See, this replication is with respect to both the prokaryotes also and the eukaryotes also. So most of the time you will find such two types of replication in eukaryotes and prokaryotes in books, but I have just included both of them part at once. Okay. So I have already conducted the lecture or detailed lecture on this point in my paid batch. So let's have a look on this replication part. So let's start with the DNA replication in case of the prokaryotes. Okay. So where this DNA replication actually begins, this DNA replication begins at the replication origin. Now what is this replication origin? This replication origin is nothing but a part of the double stranded DNA, which is at where entire replication of that DNA is going to be started. Okay, so such region of that DNA is called as what? It's called as this replication origin. Now, when this replication origin or when this replication start at this point, then it forms such structure, a two Y-shaped structure. Okay, here is a Y also shape structure, here is a Y structure, and on both of these Y-shaped structure, the DNA synthesis occurs. Okay, so that's why these are these Y-shaped replication forks. So as this looks like a fork, okay, so that's why these are also called as what? Y-shaped replication forks. And on this replication origin site only, there are origin recognition complex, means ORC, means other proteins which are actually helpful for this entire replication are going to sit. Okay. Here only this entire the machinery which is actually synthesizing the proteins will sit at the first. This will be acts as a launching pad for this. Okay, I will just announce this lecture in the group first. Okay. Okay, so that now everyone will know about this lecture. So this is about this origin recognition complex. So this is the complex of proteins which is going to be formed at this point of origin. Okay, at this point of origin. Now let's see what's the next thing. See, now whatever this protein machinery is there, which is actually synthesizing all of this replicated DNA or the new DNA, those protein complexes or that protein machinery are combinedly called as what? Replication. And this replication will sit at this point and will start to synthesize entire DNA. Next thing which are required in this DNA replications are the RNA primers. Now what is the RNA primer? Suppose this is the strand of that DNA. This is where this origin recognition complex is, or this is where this origin of this DNA is replication is going to be start. Okay, so this will form a structure like this. What will happen? See, the actual or the main enzyme which is synthesizing this new strand of DNA is the DNA polymerase. Okay, will be what? DNA polymerase. But this DNA polymerase cannot synthesize this daughter strand DNA on this fork directly. It will require some primers to get attached with. Okay, means this DNA polymerase requires some proteins which will help to get them attached with this DNA strand. So for that attachment of this DNA polymerase with these three, these are the two parent strands, this one and this one. Okay, and these whatever the new strands are going to be formed, they are called as what? Daughter strand. But before synthesizing that daughter strands, this DNA polymerase is obviously going to synthesize those daughter strands. But to attach this DNA polymerase to this parent strand, we require an enzyme or a protein complex. That proteins are what? RNA primase or these RNA primers. And these RNA primase enzymes are forming the RNA primers. Now, what are these RNA primers? The these are RNA primers are nothing but the fragments of RNA nucleotides, or you can say them as the short RNA strands. Okay, what will this RNA primase or the DNA primase will do? RNA primers are synthesized by this DNA primase enzyme. So what this DNA primase will do? It will form this RNA primers. Now RNA primers means what? These short strands of RNAs. Okay. Now why these short strands of RNAs are required? To these short strands of RNAs, these DNA polymerase is going to get attached. And once it get attached with the help of these RNA strands or RNA primers, then these RNA primers which eventually get degraded and ultimately this DNA polymerase will replace them with the help of what? DNA nucleotides. So that's why at last it is forming a daughter DNA strand. But remember, this RNA primase or the DNA primase or the RNA primers are required to be formed on this parent DNA strand before the actual DNA strand synthesis. Now there are two things. See, this is the parent DNA strands, okay, which are opened up. All right. Now there is a thing that is the concept of leading strand and lagging strand. We will see now what are these leading strand and lagging strand. Okay. Now whatever this 5 dash and 3 dash I am drawing, what are these 5 dash and 3 dash? These are the ends of the DNA. See, this entire DNA has a particular structure. In that, whatever the structure of the DNA is, it is having a one end. We can call it as 5 dash end. When we will call it as 5 dash end, at the 5 dash end, a phosphate PO4 group will be present. At the end, why it is called as 5 dash phosphate? Because this phosphate will be attached to the 5 dash structure or 5 dash position of this pentose sugar in this DNA structure. We will see after this DNA structure also. So this is the case because this 5 dash group is attached to the 5 dash carbon. So that's why it is called as what? 5 dash group. And to the 3 dash end, this hydroxyl group is attached. So that's why the another end of this DNA is referred to as what? 3 dash. And which is that group which is at this 3 dash end, that is called hydroxyl group. Okay. So because of those different groups at different position of the DNA, these are the different ends of the DNA, 5 dash end and 3 dash end. But remember always at the 5 dash end a phosphate group will be present and at the 3 dash end a hydroxyl group will be present. Same case in this case also there will be a 3 dash end and 5 dash end will be there. Okay. Sorry, I have just made by mistake here. So as this end is 5 dash end. Okay. So this end would be what? This end would be 3 dash end. Okay. I will just rub the entire annotation so that it will be easy for you. Okay. So let's draw. This is the DNA strand which has been just been opened. Okay. As every other DNA strand, this will also have a 3 dash end and 5 dash end. Okay. What I am going to explain you is the concept of what? What is the leading strand and what is the lagging strand. Okay. And all of these DNA strands are what? Complementary to each other. Means suppose this is the 5 dash end, the other end will be 3 dash, and if this is the 3 dash end, the end of the DNA which is complementary to it will be what? 5 dash end. Same case will be this will be. As this is the 5 dash end, this end will be the 3 dash end. Right? Now when this replication 4 gets open, now the DNA polymerase is going to synthesize DNA on these two strands. Okay. But this DNA polymerase has a specific characteristic that it can only synthesize the daughter strand in this 5 dash to 3 dash direction only. Okay. Means whatever this strand is going to be formed from this DNA polymerase, it can only form a continuous strand in this 5 dash to 3 dash only. It cannot travel. Now as this is the 5 dash to 3 dash direction, this direction would be what? This direction would be what? 5 dash to 3 dash direction. This one. Complementary to this other. Complementary to this. But the thing is that whatever this newer strand is getting opened, that will not open at all. Now from here, this DNA polymerase can synthesize this entire DNA up to here. Okay. But what will happen that now the next thing would be what, this new part of this DNA strand is going to be open. When this strand gets open, we will get these newer strands, right? So this DNA polymerase which has synthesized this fragment 5-3' now again have to travel back on this lagging strand. On this strand, what will happen? This DNA strand, this DNA polymerase is synthesizing this strand in this direction, 5-3' direction. Now it can continue in that direction only, because in 5-3' direction it can only synthesize. So it can continue to synthesize the leading strand. But on the lagging strand, it will have to travel back to again this end, 5' end. Then it can again synthesize this 5-3' direction. So that's why there is one strand is lagging strand and one strand is leading strand. So the thing is that RNA primers which are actually required for this DNA polymerase initiation of this DNA synthesis are required only for once on the leading strand. But on the lagging strand, as this DNA polymerase has to travel back and attach to the newer 5' end, so multiple primers will be required on lagging strand. Okay, multiples will be required. Okay, so that is the thing with the case of this RNA primers. Now this is the how this RNA primases are synthesizing this actually primers. This is the single stranded DNA is there. To this primase enzyme gets attached and it synthesizes the complementary strand of containing RNA primers. Okay, or RNA nucleotides like adenine, guanine, cytosine, uracil. Okay, these are the RNA primers. Now you can see these are in also which direction? 5-3' direction. Okay. Now let's see what are the different DNA polymerases that are involved in this prokaryotes. Okay, DNA replication in the prokaryotes. These are different DNA polymerase or different machineries work on leading strand and lagging strand. On leading strand, these nucleotides are added directly to as this leading strand is 5-3' direction. Okay, so these nucleotides can be added to this continuously. Okay, and that addition will be helped with the help of what? DNA polymerase. And even if on this leading strand DNA polymerase makes some faulty addition of any nucleotide, this DNA polymerase can itself correct this or it can displace this faulty pairing, and it can just add this back to its position. Okay, so this is called as proofreading. So that can be that is one of the characteristic of what? A DNA polymerase. So now let's see what happens on the lagging strand. See, on the lagging strand, as we have seen, this DNA synthesis is going to be discontinuous. So these are different enzymes that are acting. What is the role of these nucleases? Nucleases, then RNA, repair DNA polymerase and DNA ligase. Now, what this nuclease will do on this strand? On suppose this is the lagging strand. Okay. See, these RNA primers now needs to be what? Needs to be removed. Okay. These are synthesized by what? These are synthesized by the primase. So this nuclease will remove this RNA primers, and this DNA or this repair DNA polymerase will replace these fragments with the help of DNA nucleotides. So the now RNA primers were now replaced by the DNA fragments. Then the last enzyme that will come, that is called as DNA ligase. Now what this DNA ligase will do? DNA ligase will replace this, or DNA ligase will connect these DNA fragments and form a continuous strands even on the lagging strand. Now whatever these continuous strands of DNAs are, they are called as what? Okazaki fragments. Okay. Just consider most of them are consisting of what? DNA fragments only, right? So as they are DNA fragments, they are called as what? Okazaki fragments. Now we will going to see the next part. Okay, what is the next part is that the proteins, which are the proteins which are acting at this replication fork. So let's see one by one. Now what is the replication fork? This is the replication fork. Let's see what are the proteins which are acting at this replication fork. The first one, that is DNA helicase, it is just opening this entire replication fork. It is a DNA helicase. Next is single stranded binding proteins. Now, what are the single stranded binding proteins are? See, whenever these strands are getting open, there is a requirement of single stranded binding proteins which will protect this single stranded DNA from attachment with any other protein complexes. Okay, so that's why this single stranded DNA binding proteins immediately attach to this lagging strand. Okay, in the case of leading strand, as this DNA polymerase is continuously present at there, so that will be required no much requirement of this single stranded binding proteins. But on this strand, on this lagging strand, it will be required, right? The next is DNA topoisomerase. Okay. Now, what this DNA topoisomerase is going? Suppose, see, this is the DNA strand. Now, whenever this DNA helicase is unwinding them, so what is going to be happen is that after a particular winding, there will be structure which are called as supercoil structure. See, this is the tension because of this uncoiling. Whenever this DNA helicase is uncoiling this DNA strand, at that time, because of that uncoiling, at the last, there is a torsional tension gets produced because of that uncoiling. So that is torsional tension. It is because of the unwinding of the DNA strand. Okay, so this torsional tension is actually needs to be get what needs to get released. Okay, relaxed. So that tension is released with the help of topoisomerase. So what this topoisomerase will do? This topoisomerase will make a cut on this DNA strand, so that whatever the next the next shape of this DNA will be seen like this. Now this is what comparatively comparative to this DNA is now less tensed. Okay, so that tension release or that release of that torsional tension is going to given by this DNA topoisomerase. Next, then is the sliding clamp. Now, what is this exam that is called a sliding clamp? See, this sliding clamp is nothing but which is actually helpful for the DNA polymerase attachment with the this leading strand and lagging strand. So this is the sliding clamp which is actually holding this DNA polymerase on this strand. Clamp loader is what? Clamp loader will be the another protein which is actually loading this sliding clamp on this DNA strand. Okay. Now we will see. See, this is the actually the diagram of this entire replication fork is right. See, you can see on this leading strand, this DNA is getting continuously synthesized with the help of this DNA polymerase. This is what? This is the sliding clamp. Okay, which is holding this DNA polymerase. On the lagging strand, you can see this is the old fragment of Okazaki fragment. This is the newer strand which had just this DNA polymerase had synthesized. Now this is the before that, as this newer strand got exposed, this immediately this single stranded binding proteins attaches with it, and this RNA primates are synthesizing the RNA primers on it. Now what will happen next? This entire DNA polymerase will shift from here to this point, and this primase will get displaced. So this will how this entire cycle will happen, and this entire lagging strand also get the DNA strand continuously. Okay. Now these are the proteins we have already seen. Now let's see in the case of what happens in the eukaryotes. What happens in the case of replication in eukaryotes? Okay. So topoisomerase 2 releases tension. Yes, there are different types of topoisomerases are there. Okay. That will come. Okay, so let's see these five distinct DNA polymerase in eukaryotes. As I say, see this, there are many particulars, many enzymes are there, but to just enlist all of them in this shorter YouTube sessions won't be possible. Okay, so that's why this is the shorter version of that lectures, and a much detailed version of these lectures obviously are there for the paid batch students. Okay, so this is the third part. Okay, this is the second part, that is replication in eukaryotes. So let's see how it's begin. Okay, so in case of this replication in eukaryotes, whatever this origin recognition site are, these are two sites where this replication can start. In case of these eukaryotes, these sites are what? Ori beta and Ori gamma. Okay, and at these points, these are the possible sites of DNA replication. Okay, at this DNA replication can initiate. So in case of this eukaryotes, at here this pre-replication complex get seated, and which is the enzyme that will just unwind this entire strand like this. It is called as CMG helicase. Okay, so this would be the CMG helicase which is unwinding this entire strands. Now there are different types of polymerases are involved in these eukaryotes. They are like DNA polymerase alpha. Okay, and what is the function of this DNA polymerase alpha? It can work on both the leading strand and the lagging strand. Okay. Then this DNA polymerase beta is there, which can just repair the DNA. Okay, if there are some conformations or misadditions are there on this DNA, so that repair can be occurred with the help of DNA polymerase beta. Then there is DNA polymerase delta. Okay, which is actually working on which strand? Leading strand. It is also having proof reading activity. Means what? It can correct these wrong additions of the DNA nucleotides. Then there is this DNA polymerase eta is there. What it is doing? It is mainly working on this lagging strand. It can also proof read, means it can also correct the wrong nucleotides that has been added. Now let's see what is this fourth, that is the primase initiate the DNA synthesis. Means as in the case of prokaryotes we have seen, we also require the primase here also. So this primase on the leading strand will be required for the ones same as the prokaryotes. Then this sliding clamp. In case of eukaryotes, they are referred to as what? PCNA, means what? Proliferating cell nuclear antigen. Now what this PCNA will do? This PCNA will just recruit this DNA polymerase delta on this leading strand. And there is a clamp loader is there, as we have seen in the case of prokaryotes. In case of eukaryotes, it is specifically named as what? RFC, replication factor C. So this will be what happened on the leading strand. On the lagging strand, these single stranded binding proteins we have seen in the case of prokaryotes, they are called as what? Replication protein A. There are just specific names in case of eukaryotes are there. Okay. So obviously the next enzyme, see, once these these RNA primers are formed, they are going to be removed by which? By this nucleases. So the next enzyme will be what? Nuclease. Now let's see one by one the proteins on this replication fork in case of eukaryotes in detail, right? One by one. The first is DNA helicase, CMG helicase in case of this eukaryotes. Okay, it will just open up this entire strand. The next is replication protein A, RPA. What are these RPAs are doing? They are just get attached to the DNA strands exposed by the helicase and prevent these strands from reforming the complementary base pairs. Means what? Whatever these strands are separated. Okay, if we just don't protect these strands from again forming a structure like this, okay, then we will have to protect these, right? For that protection is occurred with the help of this RPA, single stranded binding proteins. What they will do, they will not allow these two strands to get attached again and to form a structure like this, or just to reform the bonds in between them. So that will be prevented by what? This single stranded binding protein, or specifically in case of eukaryotes only, it is called as RPA. Then this is the DNA topoisomerase is the same as we have seen in the prokaryotes. Just it is named as DNA topoisomerase beta. What it will do? It will just cuts this. See, this is the initial suppose structure of DNA is there. Okay. What will happen after this opening? A part of this DNA will get opened like this. Okay. But the remaining part will get very super coiled like this. Okay. Now this is very super coil structure. Now this entire DNA helicase cannot progress beyond this, because this is a very coiled structure is there right now. So that coiling is removed by this DNA topoisomerase beta. Okay, this is removing what? Excess twisting tension. Then this processivity factor, the sliding clamp is this PCNA in case of what? Eukaryotes. Then this RFC is what? Recruiting that sliding clamp. Then the telomerase is what? Now there is a thing with the case of this telomerase, that is what? See, what happens usually is that, see, suppose these are the RNA primers, right? Okay. So this DNA ligase can join this Okazaki fragment, and just this nucleus can remove this RNA primers. But what will happen is that this DNA ligase can only fill gap at this point only. It is not able to fill the gap at this point only. Okay, so that's why it is a vacant position. And why this DNA ligase cannot fill the gap at the end part or the terminal part of DNA is because it requires two adjacent Okazaki fragments to get attached. Okay, when the DNA ligands is attaching, it is also filling a gap in between these two. Okay, so that's why at the end what will be happen, only one phi dash end of this Okazaki fragment is available. As there is no second end of this next Okazaki fragment is available, this DNA ligase cannot fill this gap. So to fill or to replicate these end parts of this DNA, there is another enzyme that is called as what, DNA telomerase is there. Now what this DNA telomerase is doing, DNA telomerase will replicate the end strand of DNA. So just completing this entire DNA replication in the eukaryotes. Okay. So this was the case with this replication, the DNA replication in case of the prokaryotes also, and DNA replication in the eukaryotes also. If you have any queries on this part, you can ask in the comment box. I will attend. Otherwise, we will stop our session here. Okay. You have any queries? कुछ डाउट है क्या? Okay, and we are continuously to have such sessions, right? So even if they will be short, but we will have a more continuous sessions. Okay, so I don't see any doubts are there. So we will stop here and we will meet for next session with the next chapter, mostly the transcription. So till then, have a great day. Good night.